Hey everyone, welcome to this new video. Let me show you some pictures. So what you're seeing on the screen, these are real patients with T and B cell lymphoma. What you're seeing here is what happens when cells forget how to die. The swollen lymph nodes, the organ infiltration, the complete breakdown of normal immune function. It's devastating, and unfortunately for many of these people, it's fatal. But here's what's fascinating. Almost all of these cases trace back to this tiny gene called PIM1, which is a kinase that's supposed to help cells survive stress. But when it goes wrong, it turns our own immune cells into killers. And that's just the beginning of our PIM1 story. Let's start chasing this gene around the globe using our biobanks. Okay, so let's start with our East Asian biobank, starting with China Kadoorie Biobank. When we look at PIMS1 associations in the vast China Kadoorie Biobank, the immediate signal isn't a disease, it's a protein quantitative trait locus, or PQTL. And I say this because this biobank does have diseases and phenotypes as well. It's not just a PQTL biobank. And as you can see, the strongest hit here is a robust association with PI16, which is peptidase inhibitor 16, and the p-value is 4.6 times 10 to the minus 11 across only 4,000 samples, right? So this isn't just a random correlation. PI16 is a critical protease inhibitor known to be involved in modulating fibrosis, cardiovascular health, and T-cell regulation in autoimmune context. So the subtle yet crucial connection here is that a genetic variant near PIM1 is influencing the level of a key regulator of the extracellular matrix and inflammation. This immediately puts our target PIM1 at the nexus of not just a specific disease, but the underlying inflammatory and tissue remodeling processes that underpin them, a much broader therapeutic window. Moving on, South Korea's KOJES. Okay, so here we see the narrative solidify, but it shifts slightly into the hematopoietic, into the hematopoietic system. Here, the top hits are for classic blood cell traits, specifically platelet count and hematocrit, both with p-values in the 10 to the minus 8 and 10 to the minus 6 range. This data point, derived from a meta-analysis with the Biobank Japan data, provides crucial independent replication, reinforcing the idea that PIM1 variation impacts blood cell line regulation. And for drug development, this is a profound clue. PIM1 is known to be a regulator of cell survival, so seeing it linked to platelet and erythrocyte counts suggests its biological influence extends deep into myelopoiesis and erythropoiesis. So targeting PIM1 could therefore have systemic hematological consequences, a key consideration for adverse event profiling in any clinical trial that we want to do. And if we check out Biobank Japan itself, the Biobank Japan data with its larger sample size, here is where we go from cell traits to complex diseases. While platelet count remains the top hit, we now see direct explicit links to prostate cancer and autoimmune disease, both with strong p-values near 10 to the minus 7. So this is like the pivot point here. And the fact that the same genetic locus associates with both a blood cell trait and a common solid tumor, prostate cancer, and a complex autoimmune disease, this tells us that this is not a narrow mechanism. It suggests that PIM1 is a shared vulnerability, likely driving uncontrolled cell proliferation in the case of cancer and dysregulated lymphocyte survival in the case of autoimmunity. This positions PIM1 as a pleiotropic drug target, meaning its modulation could have simultaneous therapeutic effects across multiple, seemingly disparate conditions. Moving on, we have Taiwan Biobank. So the data from the Taiwan Biobank offers another layer of tissue specificity, which is the liver. We see significant associations with SGOT and the fibrosis 4 score. So SGOT is, or like SGOT, this one, is a liver and heart enzyme doctors check in blood tests to see if your tissues are damaged. And the FIB4 score is a validated, non-invasive indicator of liver fibrosis and scarring. And I want to bring your attention to the link between the initial PI16 finding, which I showed you here, which is a fibrosis modulator, and this explicit liver fibrosis signal. So the systemic theme is now inflammation and tissue repair gone awry. A PIM1-driven mechanism impacting immune survival could be a driver of the chronic inflammation that eventually leads to fibrosis and subsequent cirrhosis. High-stakes targets for drugs aimed at mitigating end-stage liver disease. And now let's check out MVP East Asian cohort as our final East Asian Biobank. And here, finally, we confirm this liver and digestive system focus with the MVP East Asian cohort data. The top signals are for clinical codes, chronic liver disease and cirrhosis, and symptoms involving the digestive system, with p-values around 10 to the minus 5. And again, this moves us from a biomarker, which was FIB4 score, to an actual clinical outcome, which is cirrhosis. Okay, so that was our East Asian biobanks, and moving on to other parts of the world. So to transition from the East Asian cohorts to the broader global landscape, we started here. Let's start with the Qatari Biobank as our bridge to Europe. Okay, so our only Middle Eastern biobank, the Qatari Biobank here, the PIM1 associations maintain the theme of systemic health, but shift focus to the metabolic and coagulation axis. The top hit is for prothrombin time, an essential marker of the extrinsic coagulation pathway, alongside hits for LDL, cholesterol, and triglycerides. This is a critical nuance. While our previous data hinted at hematopoiesis, with the platelet signal, this finding here implicates PIM1 variation in hemostasis and lipid metabolism. So why does this matter for drug design? Because it suggests that PIM1 activity is integrated into the complex interplay between inflammation, lipid flux, and vascular health. And if we develop a PIM1 inhibitor for cancer, for example, we must meticulously monitor its impact on a patient's coagulation profile and dyslipidemia, potential high-risk adverse events. Let's go to our next one here, Biobank Russia. We're making our way into Europe, the Eurasian context with the Biobank Russia data. The strongest signals are unambiguous. They center on high C-reactive protein, or CRP, which is a gold standard marker of systemic inflammation, with p-values below 10 to the minus 6. This confirms that the PIM1 locus is a major contributor to chronic low-grade inflammation across a diverse set of global populations. And the fascinating and somewhat sobering observation here is the association with death from other reasons, not cancer or cardiovascular diseases. And while speculative, you can see that this phenotype has only 38 cases, but somehow it reached like a I would say, good p-value of 10 to the minus 5. I mean, it's still not the genome-wide significant, but with only 38 people, that's like very good. So again, like while speculative, given the inflammation theme, this is a flag that PIM1 driven biology may contribute to the general aging process or broad systemic failure linked to chronic inflammatory states. And for a drug target, this is a strong hint that PIM1 intervention could potentially affect health span, a potent driver for therapeutic pursuit. Let's check out FinGen next, which is not that far. So the thing about the FinGen data is that this whole biobank is a genomic powerhouse, and it provides the highest resolution of PIMS1, autoimmune, and inflammatory pleiotropy. We move from broad traits to highly specific clinical entities. So note the strongest enrichment for spine-associated autoimmune and inflammation-driven conditions here. And if we look closely at the p-values, many of them are below 10 to the minus 19, marking this as an extraordinarily strong replicated association in the European context. And critically, we also see hits for type 1 diabetes, which is interesting, and multiple sclerosis, MS. And here the common denominator is T-cell mediated autoimmunity. Now here's the thing, PIM1 is a key kinase downstream of the JAK-STAT pathway. The JAK-STAT pathway is a major driver in T-cell differentiation and survival. So these FinGen results provide in vivo human genetic validation that PIM1 dysregulation is a central shared mechanistic vulnerability across the spectrum of HLA-B27 associated and other severe autoimmune diseases. And if we go back to FinGen, so this immediately suggests a high-value opportunity for drug repurposing if a successful PIM1 inhibitor already exists. Let's check out UKB. Finally, the massive scale of the UK Biobank brings us back to high-impact clinical outcomes. The top association, 5 times 10 to the minus 8, is with primary pulmonary hypertension. This is a rare, severe, and devastating disease characterized by uncontrolled proliferation and survival of pulmonary arterial smooth muscle cells, a textbook example of PIMS1 pro-survival function in the vascular system. The simultaneous association with secondary malignancy of respiratory organs reinforces the oncogenic potential of this pathway, and these findings, when combined with the coagulation and lipid data from Qatar, paint a picture of PIM1 as a fundamental mediator of cell proliferation and survival in 3 distinct yet interconnected systems. 1, hematopoietic, immune, 2, liver, metabolic, and 3, vascular, oncogenic. And this profound pleiotropy is the core of PIMS1 attractiveness and its challenge as a drug target. Let's go to America. And let's start with A2F. Before synthesizing the global findings so far, we encounter an important piece of context from an association filtering tool, the A2F knowledge panel. And as you can see here and on this table, interestingly, despite the peripheral vascular and lipid signals we observed earlier, this specific tool finds no evidence for a vast array of core cardiovascular phenotypes like myocardial infarction, CAD, or hypertension. This apparent contradiction isn't noise, it's a signal. It suggests that the PIM1 locus's effect on the vascular system might be tissue specific, like the pulmonary hypertension hit we saw, or it may act via intermediate phenotypes like inflammation and coagulation rather than directly driving the complex multifactorial common vascular diseases shown here. So for drug developers, this suggests that systemic cardiovascular risk might be lower than initially feared, but careful monitoring of specific vascular beds is still paramount. Our next biobank, MGI BioVU, derived from electronic health records, revalidates our core hematological and inflammatory themes. We see the familiar strong signal for platelet count, which is, by the way, this is how I found this gene. And also PTT, partial thromboplastin time, which is a measure of the intrinsic and common coagulation pathways. But look at the last one, amylase. Amylase is a key digestive enzyme, and its presence in the blood, often due to pancreatic damage, links PIM1 variation to gastrointestinal or pancreatic inflammation and damage, a critical extension of the liver and digestive signals we saw in the East Asian and FinGen data. Now let's turn to our Canadian biobanks, starting with Cartegen, which is our French-Canadian biobank. So the French-Canadian cohort links PIM1 to allergy and cancer, maintaining the autoimmunity proliferation theme. But in the white Canadian cohort, CLSA, a completely new signal appears, a highly significant association with QT interval, a measure of cardiac repolarization. This is a high alert clinical connection. Drugs that prolong the QT interval can cause potentially life-threatening complications. So for a PIM1 inhibitor, this means a thorough cardiac safety assessment will be non-negotiable. And I'll show you this towards the end of the video, where we discuss some of the clinical trials for PIM1 inhibitors. And I'll show you there that one of the things that they take into account when enrolling people in these clinical trials is cardiac safety. So the QT signal suggests a direct effect on myocardial ion channel regulation, entirely separate from the vascular effects, which requires a distinct toxicology approach. And finally, our heavyweight dataset, MVP. So I'm just going to start with MVP meta, which is like all the ancestries combined. The MVP meta-analysis combining all ancestries provides the statistical hammer of global validation. The strongest associations with p-values down to 10 to the minus 15 are no longer with platelets, but with monocyte fraction. Monocytes are critical innate immune cells that differentiate into macrophages. This is the genetic evidence that PIM1's primary function in human disease is rooted in innate and adaptive immune cell regulation. So this meta-analysis ties everything together. Strong signals for monocyte and platelet count, reinforcing the immune and hematological axis. Strong associations with total cholesterol, which is related to the metabolic axis, and explicit disease links to prostate cancer and lesion of the stomach and duodenum, which is the proliferation GI axis. Now let's do ancestry-focused MVP2, starting with MVP African ancestry. So examining the ancestry-specific MVP cohorts is crucial for confirming that this is a global mechanism, not population specific. The African cohort here shows the strongest hit on platelet count, maintaining the hematological theme, and introduces a clear association with height, which is interesting. If you also watched the PIM1 video, this is, starting to sound familiar. Next up, we have MVP at mixed American ancestry, and the at mixed American ancestry cohort pivots to disease, showing a link to type 2 diabetes with neurological manifestations, adding a specific nervous system complication to the metabolic picture. And MVP, we did East Asian, let's do European. And so finally, the European cohort here presents the strongest signal of all, which is monocyte fraction at an extraordinary 1.5 times 10 to the minus 21, alongside multiple hits for MCH and MCV, classic erythrocyte indices. This confirms PIM1's profound and multilineage influence on the hematopoietic system. Okay, so the PIM1 locus is now definitely established as a central regulator of hematopoiesis, inflammation, autoimmunity, and proliferation, across all major ancestries. And the challenge is now moving from association to mechanism. This level of pleiotropy confirms PIM1 is a target of immense therapeutic potential, but requires an extremely precise drug design to manage systemic off-target effects. We now zoom into the gene itself to understand the fundamental mechanism driving these associations. Okay, PIM1 is located on human chromosome 6p21.2. Let me show you. So these are all the human chromosomes, and if we go to the P arm of chromosome 6, and the band was 21.2. So the gene is somewhere here. And the thing that I want to say here is that 6p21, which includes all these bands, is where most of the HLA genes are. So this little square here is the most gene dense area in the human genome. This is a small region, but it definitely punches above its weight. Now, let's talk about the PIM1's interesting backstory, because it didn't even start with humans. It begins with viral leukemogenesis in mice. So in the 1980s, researchers identified PIM1 as a highly conserved cellular gene frequently activated by proviral integration. This early work immediately flagged PIM1 as a proto-oncogene, which means that it's a normal gene that, when deregulated, could drive cancer. The initial characterization showed PIM1 encodes a protein kinase with no transmembrane region, suggesting its function is entirely intracellular signaling. And crucially, they found its expression was primarily restricted to B lymphoid and myeloid cell lines, the very cell types we now know are central to the autoimmune and hematological signals we just explored in the Biobanks section. So this discovery provided the first critical clue for drug development, which is PIM1's physiological role is rooted in hematopoietic development. Its presence is high in fetal hematopoiesis, and its overexpression is a hallmark of acute leukemias. So targeting it, therefore, is an attempt to exploit a highly specialized cancer hijacked mechanism. Then, a key finding was the demonstration by Chris Saris and colleagues that PIM1 function as a protein serine threonine kinase. So they showed the gene actually encodes two distinct protein isoforms. And remember, this is like before all the ensemble data sets and all the transcript tables that I'm about to show you in a second. So they discovered that this gene actually encodes for two protein isoforms, and these are synthesized via alternative translation initiation at an upstream CUG codon. So this use of a non-canonical start codon to produce different isoforms with potentially different subcellular localizations or functions is a sophisticated, subtle regulatory mechanism we must consider when designing inhibitors. And from a therapeutic standpoint, we need to know how fundamental is this gene. To answer that, let's turn to comparative genomics. All right, so this panel from Open Targets, which actually comes from Ensemble, but this comparative genomics lens confirms that PIM1 is not an isolated target. It belongs to a highly conserved small family of kinases. The first key result here is the paralog data. Human PIM1 has two close relatives. We have PIM3 and PIM2. So PIM3 with nearly 70% sequence identity and PIM2 with 54% identity. This is an important detail because any drug designed to inhibit PIM1 will likely exhibit off-target activity against PIM2 and PIM3. Now, this is not necessarily a bad thing. In fact, in many cancers, dual or pan-PIM inhibition is the desired mechanism. However, it mandates careful preclinical work to understand the systemic functional consequences of inhibiting the entire PIM family, given their roles in different biological contexts. Next, the ortholog data across species from mouse and rat to rabbit and pig shows remarkable conservation with identity scores consistently above 92. Even the zebrafish ortholog retains over 70% identity. So this high degree of evolutionary conservation is our strongest evidence that PIM1's function is essential and has been under severe purifying selection for hundreds of millions of years. For drug development, this is a green light for utilizing well-established genetically modified animal models like the knockout and transgenic mouse models mentioned in the gene's history, as their PIM1 function closely mirrors our own. So this is good news for us. And if I scroll up for a genetic constraint panel from Nomad, so if evolutionary history suggests the gene is important, this genetic constraint data from Nomad tells us precisely how intolerant human populations are to damaging mutation in PIM1. This is a crucial metric for drug developers as it predicts the functional resilience of the target. The data shows a striking signal of constraint. So one, for synonymous mutations which don't change the amino acid, the observed to expected ratio O to E is 1.05. This is essentially neutral as expected. For missense mutations, and these are mutations that change the amino acid, the OE, the O to E observed to expected ratio, all of a sudden plummets from 1.05 all the way to 0.627, which means we observe only about 63% of the missense mutations we expect to see in the population. And the Z-score of 2.85 indicates significant depletion. And the most compelling signal is for protein loss of function. So for protein loss of function mutations, where the observed to expected ratio is a drastically low 0.252. We only observe a quarter of the expected crippling mutations. This results in an extremely high PLI score of 0.998 and a very high constraint assessment. So the implication for drug design is that a high PLI score signals that PIM1 is intolerant to haploinsufficiency, meaning that the loss of even one functional copy is poorly tolerated in humans. This is a profound therapeutic insight, as PIM1 is an addiction gene for the cells that rely on it, most notably cancer cells and specific immune lineages that we're interested in targeting. So therefore, an effective PIM1 inhibitor that chemically mimics a loss of function mutation should ideally be extremely effective in diseased tissues that have become addicted to PIM1's pro-survival function, while potentially minimizing harm to normal tissues, which are highly sensitive to its reduction but have other backup pathways. Now let's check out the RNA and protein expression data. We now move to the baseline expression profile of PIM1 across human tissues. And this data is critical because it tells us where the PIM1 protein is physiologically active, thereby confirming the source of our GWAS signals and informing the likely tissues for on-target off-tumor toxicity. And looking at the RNA expression atlas on the left, the distribution is not uniform. The highest expression is clearly concentrated in the hematological and immune systems. Blood is highest, followed by esophagus, immune organ, and mucosa. This strongly confirms our GWAS findings linking PIM1 to blood cell traits and a wide range of autoimmune and inflammatory diseases. The fact that blood and immune organ are the highest sites of RNA expression means that any PIM1 targeted drug will have its highest steady state concentration of target in these cells. And this validates the efficacy potential in hematological malignancies and autoimmune conditions. But it also necessitates intense monitoring for myelosuppression and immune function changes in clinical trials. Now, let's look at the protein expression. So the right panel here from the Human Protein Atlas, HPA, the protein data generally mirrors the RNA, confirming high protein expression in immune organ, spleen, lung, and intestine, further validating its role in immunity and inflammatory lung disease, like the secondary malignancy hit that we had, and gastrointestinal conditions. But I want you to take note of the differences between the RNA and protein data. So for instance, blood has the highest RNA expression, but is listed as NA for protein, which often points to methodological challenges in isolating specific cell types for the HPA assay, or extremely tight post-transcriptional control. Conversely, brain here shows relatively low RNA but moderate protein expression. This is a subtle but important detail. It suggests PIM1's kinase activity in the central nervous system may not be exclusively transcriptional, and its role in neurological phenotypes like the type 2 diabetes neurological manifestations we saw warrants deeper investigation, perhaps concerning BBB penetration for any drug candidate. And BBB stands for the blood-brain barrier. So in sum, the expression profile establishes PIM1 as a master regulator of the immune hematopoietic mucosal axis. Any therapeutic targeting must be designed with the knowledge that these are the tissues most heavily reliant on PIM1 for baseline function, requiring a careful balance between efficacy in disease and acceptable systemic toxicity. So, we now dive into the PIM1 locus itself, located on the forward strand of chromosome 6. This view reveals the complexity underlying its functional pleiotropy. The landscape is dominated by multiple transcripts and a dense, dynamic regulatory architecture. The gene is a lesson in isoform diversity. We are dealing with 8 splice variants, 6 of which are actively protein coding. The primary main select transcript PIM1 201 produces 313 amino acid protein, but critically other coding transcripts exist, generating isoforms that vary in length. We see 318, 277, and even 237 amino acid long proteins. And recall the historical anecdote about PIM1 producing 2 protein products from alternative translation initiation. This paper that I just showed you, this one. So this transcript map provides the modern high-resolution context. Multiple transcripts coupled with alternative initiation is the molecular explanation for how PIM1 can exert different functional effects in different tissues despite being the same gene. An isoform expressed in a monocyte may have slightly different domain usage or stability than one expressed in a prostate cell, providing distinct therapeutic windows. If we go back to Ensemble, and if I scroll down for the regulatory features around the gene, so the regulatory features track is where the GWAS signals truly connect to function. We see a highly segmented and active regulatory region evidenced by the clustered features. We have promoter flanks, we have enhancers, and also CTCF sites. Yeah, and look at the EMAR regions. And as you can see here, EMAR is epigenetically modified accessible region. This indicates areas of open chromatin that are actively accessible for transcription factor binding. The GWAS hits we observed in the global biobanks are almost certainly mapping to these non-coding regulatory regions. A single nucleotide polymorphism at an enhancer site here could subtly shift the binding affinity for a tissue-specific transcription factor, say an immune-specific factor, causing a slight up or down regulation of the PIM1 transcript only in that specific cell type, like monocytes. So this single regulatory hit would then propagate all the associated phenotypes we saw, like monocyte fraction, autoimmune disease, and inflammatory markers. So this regulatory density reinforces the difficulty and the potential also for drug development. The challenge isn't just inhibiting the kinase, it's understanding which regulatory elements are driving overexpression in which disease, allowing for therapies, perhaps using antisense, oligonucleotides, or gene editing even, that target the transcriptional driver, not just the final protein. And speaking of protein, well, here's the protein. PIM1 protein is a serine threonine protein kinase. This is the functional heart of our target. This is the molecule that directly executes the pro-survival and proliferative functions suggested by the GWAS data that we saw on the biobank section. And if I scroll down for the Go annotations, so for the eukaryotic cellular processes, you can see that PIM1's core activity is protein serine threonine kinase activity. Like all kinases, it requires ATP binding, as you can see here, to catalyze the transfer of a phosphate group. Interestingly, it also exhibits manganese ion binding, which, while not as common as magnesium for kinases, hints at a unique aspect of its catalytic domain or regulatory mechanism. Crucially, it's also annotated with protein serine threonine kinase activator activity. So this means that PIM1 doesn't just phosphorylate downstream effectors, it can activate other kinases, positioning it as an upstream signaling node that amplifies proliferative and survival signals. If I scroll down on this table, you can see that the biological process Go terms directly explain PIM1's oncogenic power and its role in autoimmunity. The central theme here is regulation of cell survival and proliferation. We see multiple annotations for the apoptotic process, specifically negative regulation of apoptotic process. This is the key mechanism in cancer. This is the key mechanism in cancer. PIM1 phosphorylates pro-apoptotic proteins, effectively shutting down the cell's suicide mechanism, allowing unregulated survival. And this ties back perfectly to the associations with prostate cancer and secondary malignancies. And furthermore, it's a master regulator of proliferation. And note the explicit terms regulation of hematopoietic stem cell proliferation and also positive regulation of cardiac muscle cell proliferation and also regulation of mitotic cell cycle. This validates the GWAS hits for platelet count and pulmonary hypertension and cardiomyopathy. So the interesting thing here is scientists have noticed that PIM1 gets switched on when the immune system is active, especially when signals like IL-2 and IL-6 are released during inflammation. That tells us it's part of the body's response to infection or stress, but interestingly, it's also annotated for negative regulation of innate immune response and cellular response to type 2 interferon, which means that it acts as a brake or regulator on the immune response, not just a pure accelerator of cancer. So instead of being only a gas pedal for cancer or inflammation, PIM1 may also work as a brake, helping the body strike the right balance between attacking threats and avoiding damage to itself. A particularly high value annotation for drug development is positive regulation of TOR-C1 signaling. The mTOR pathway is arguably the most critical pathway in cell growth, metabolism, and proliferation. So PIM1's ability to regulate TOR-C1 means that even a partial PIM1 inhibitor could have a synergistic effect when combined with existing mTOR inhibitors that are already clinically approved. And this opens up a clear strategy for combination therapy and drug repurposing trials. So transitioning from general function to the atomic level, we analyzed the post-translational modifications, or PTM, and structural dynamics of the 313 amino acid PIM1 protein, the main selection. And this information is the bedrock of kinase inhibitor design, as it defines the activation state and stability of the target. The curated PTM data shows 4 key phosphorylation sites, as you can see here. Now, in the world of kinases, phosphorylation isn't random, it's a switch. So these PTMs fall into 2 groups, N-terminal sites and internal sites. The N-terminal sites, these are located in the highly flexible N-terminus, phosphorylation here is often associated with regulating PIM1's stability and degradation, possibly by preventing it from being targeted by ubiquitin ligases. And internal sites, these sites are more likely to be involved in regulating PIM1's catalytic activity or its interaction with binding partners. And unlike many kinases which require phosphorylation within their activation loop, their A loop, PIM1 is constitutively active, meaning it's active without its canonical A loop PTM. But, however, its activity is amplified by these other PTMs, which primarily seem to enhance its stability and half-life. So, drug design strategy here is twofold. Target the active site directly and explore small molecules that interfere with these PTMs to destabilize the protein, thereby inducing its degradation in addicted cells. The data on curated disorder is perhaps the most fascinating from a structure-based drug design perspective. We see a defined region of intrinsic disorder from residue 14 to 31 here in the N-terminus, and critically this region is also identified as a derived binding mode disorder to disorder region, which means that it's flexible until it encounters a binding partner, at which point it assumes a stable structure. So this disorder profile tells us two key things. One, flexibility and interaction, and two, structural drug design challenge. So the flexibility thing is that the highly flexible N-terminus is the probable site for docking many of PIM1's diverse substrates and protein interactors, explaining its broad substrate specificity. And the structural drug design challenge is that kinase inhibitors typically target the rigid ATP binding pocket. So targeting this disordered region with an allosteric inhibitor, a drug that binds away from the active site, could be a more selective strategy. Such a drug could force the flexible region into an inactive conformation or block its interaction with a key binding partner, which offers a path to greater isoform or context specificity over traditional active site inhibitors. And finally, the derived missing residues data from multiple PDB structures further confirms that the N and C terminal tails are highly mobile and often not resolved in crystallographic studies, reinforcing that PIM1's regulatory domain is structurally dynamic and therefore uniquely challenging and also rewarding to target. So we conclude our exploration of PIM1's basic biology by visualizing the complete molecule through the AlphaFold predicted structure. This ribbon model based on the PIM1 201 isoform, the main select. This perfectly illustrates the duality of the PIM1 protein, a structure that is both rigid and highly flexible, right? You can notice that by looking at the colors on this protein. We have dark blue, which is like rigid, and also the IDRs on the side, and this one is like between red and blue. So the bulk of the protein colored in dark blue represents the kinase domain. This is the catalytic engine characterized by a highly ordered, well-defined structure. The folds, helices, and sheets here form the canonical ATP binding pocket, the primary target for most small molecule kinase inhibitors. and the inherent stability and order of this domain make it highly predictable and designable. However, the image also highlights the crucial non-catalytic regions. So if you notice the red and cyan tails extending from the main body, the red region's long thread-like appearance confirms its nature as a highly flexible or disordered region. And similarly, the cyan region also appears less structured and often is associated with the disorder to order transition we discussed. These flexible regions are the regulatory and interaction platforms of the protein. So the AlphaFold model serves as a final comprehensive structural guide. The rigid core, which is in dark blue, and the flexible tails, the red and cyan tails extending from the core. So this composite view is the structural basis for PIM1's function as a versatile survival promoting kinase, and it guides our discussion on its therapeutic value. The final layer of PIM1's complexity we explore here on UniProt is its subcellular localization, which perfectly explains its plytropy and broad functional reach. We see distinct locations based on the protein isoform. The primary isoform resides in the nucleus and cytoplasm, while the secondary isoform localizes to the cell membrane. So the therapeutic takeaway here is that PIM1's activity is essentially a two-front war within the cell. The primary therapeutic action against cancer and autoimmune disease must target the nuclear cytoplasmic pool to shut down survival signaling and gene expression. However, targeting the membrane-bound pool of isoform 2 could provide a vital check on upstream pro-inflammatory and proliferative signaling initiated at the cell surface. So a successful clinical candidate might need to demonstrate bimodal activity, the ability to penetrate the cell, inhibit the cytoplasmic nuclear kinase activity, and potentially interfere with the membrane-anchored signaling. So, with the complete molecular profile now established, from global GWAS association to genetic constraint, expression, structure, and localization, we can now turn to the clinical and commercial landscape for PIM1 targeted therapies. We now look at the most critical data, the history of PIM1 targeting compounds in human clinical trials. This clinical precedent from the Kimball database validates PIM1 as a drug target, but also reveals the significant challenges encountered by early drug development efforts. The initial clinical efforts for PIM1 inhibitors were highly focused on the cancer types where PIM1's oncogenic role was first discovered. And if you recall, it was hematological malignancies. So AZD1208 from AstraZeneca and LGH447 from Novartis were tested as PIM inhibitors in phase 1 trials for conditions like AML, lymphoma, and multiple myeloma. LGH447 successfully completed its phase 1 studies for these diseases, showing initial tolerability. And SGI-1776 was also taken into phase 1 for leukemia and non-Hodgkin's lymphoma. And this focus directly validates the high RNA and protein expression we saw in the hematopoietic tissues and the known function of PIM1 as a pro-survival kinase for blood cells. Then, the compound's scope was quickly expanded to match PIM1's plytropy, with phase 1 trials initiated for solid tumors and fibrotic disorders. SGI-1776 was tested in prostate cancer, directly confirming the association identified in the early 2000s. And AZD1208 had a broad cancer phase 1 trial, and LGH447 was even tested in myelofibrosis, which is a fibrotic blood disorder, which aligns with the PIM1's pro-proliferation signals in stem cells and its potential role in fibrosis, like the pulmonary hypertension or like vascular signals that we saw earlier. The most sobering data point across this entire list is the status column. Terminated, withdrawn, these two dominate the outcomes. So why did these first generation PIM inhibitors largely fail to advance past phase 1? And there are mostly three reasons. One is, of course, safety and toxicity. Two is lack of selectivity, and three, monotherapy limitations. I think it would be extremely valuable if we took a quick look inside these trials and saw what happened. And just for the sake of time, I'm just going to pick one of them. And I'm particularly interested in AZD1208 from AstraZeneca. And the reason is, so they had 3 clinical trials. 2 of them are completed and 1 of them was terminated. So let's see what happened with the trials. And the first one, AML for AZD1208 from AstraZeneca. The official record for the phase 1A, 1B study of AZD1208 in acute myelogenous leukemia, AML, is a textbook example of a genetically validated target hitting the no clear efficacy as monotherapy wall, right? So the study targeted recurrent or refractory AML patients, and this is the hardest to treat population, where PIM1 is hypothesized to be a critical survival factor for cancer cells that have become resistant to previous chemotherapies. This clinical trial was done in 2 parts. Part A was safety testing, and Part B was effectiveness testing. So Part A, they started by testing the drug at low doses and slowly increased it. And the goal was to find the highest dose people could safely take without having serious side effects during the first month. And in Part B, once they found the safest strong dose, they gave that dose to more patients to see how well it actually worked, and specifically whether it could completely clear the disease or mostly clear it even if blood counts weren't fully back to normal. So the study stopped early and will not start again. The actual enrollment size was 55 patients. This is a substantial cohort for a phase 1 trial. And primary outcome result is no results posted for the efficacy outcome, as we found in the search. The lack of objective clinical responses was the primary driver for termination. And if I show you the participation criteria, we can clearly see signs of the safety problem, the too important to block issue, and also the effectiveness problem, the backup systems issue. So PIM1 is so essential to normal body function that blocking it can hurt healthy tissues and not just cancer. That's why in the clinical trial, they excluded anyone with heart problems, diabetes, or high cholesterol, because they suspected heart toxicity, which is a common side effect for kinase inhibitors. And they also suspected metabolic side effects like messing with blood sugar and cholesterol. So in short, the drug might work on cancer, but it also risks harming the heart and metabolism. And the effectiveness problem, the backup systems, issue is also clear here that even when the drug successfully shut down PIM1 in cancer cells, the cancer didn't die. Why? Because cancer cells just switch to alternate survival pathways like mTOR. This means that blocking PIM1 alone isn't enough. So the cancer has backups. And let's leave this trial with three main lessons. One, PIM1 is a viable target. Two, pan inhibition has a narrow therapeutic window. And three, future focus must be combinatorial and selective. Now let's move on to the other trials for this molecule. And unlike the AML trial, which was terminated early due to safety concerns and lack of response at the highest doses, the global solid tumor and lymphoma study of AZD1208, they reached its completed status. So this indicates that the trial successfully executed its plan, collected all scheduled safety and pharmacokinetic data, and reported its preliminary efficacy findings. So let's check out the cancer trial for AZD1208. This is the webpage for that. Status is completed. Conditions: advanced solid malignancies and malignant lymphoma. So the primary conclusion of this completed study was that one, the best objective response observed across all 43 patients with refractory solid tumors and malignant lymphoma was stable disease. And two, there were no objective responses. This means the drug was capable of temporarily halting tumor growth in some patients, but it was unable to cause the meaningful tumor shrinkage necessary to warrant advancement into large-scale phase 2 efficacy trials. And this completes our case study on AZD1208. In short, AZD1208 from AstraZeneca proved that PIM is a target worth hitting, but not one that can be hit alone. And based on the totality of the data we've reviewed, from the PLI score suggesting essential function to the complex structural domains to the clinical failures of first-generation pan-PIM inhibitors, the future of PIM1 targeted therapeutics lies in three areas. One, combination therapy. Two, isoform selectivity. And three, allosteric modulators. And let's end the video here. Thank you so much for joining me on this journey. Each video that I make is me learning about that gene for the first time. And I'm so happy, and it gives me so much joy to make these videos. I hope you enjoy watching them and you learn a thing or two as well. If you like the video, please like and subscribe, and I'll see you on the next one.